good morning, everyone. The mountain you see on the slide is Mount Matterhorn in Switzerland, and what that has to do with 444 pyrosequencing, that's what I'm going to talk about during the next 20 minutes. So 444 pyrosequencing comes in 3 generations, and the latest generation is from 2008. And since it's high throughput sequencing, it gives us quite huge data amounts, which give us reads with up to 500 base pairs and around 1.2 million reads per run. And so the data types that we get are either shotgun reads or mate pairs. And mate pairs come as single reads with the linker sequence somewhere in the middle. I mentioned that because it's different from Illumina and Solexa. And the 2 main applications, I guess, are whole genome sequencing and metagenomics. And there's been a bunch of publications on metagenomics done with 454, and there's been a bunch of publications on error sources and how they influence metagenomic analysis. But also in de novo whole genome sequencing, a lot of people are now using 454 data. For example, University of Oslo has coordinated the sequencing of the cod genome, and they had around 44 shotgun reads and around 20 mate pair runs and around 20 mate pair runs. And so this was based mainly on 454 data. For those of you who are not too familiar with how 454 pyrosequencing works, the DNA is fragmented, and then each single-stranded DNA fragment is attached to a bead. These beads are then placed in wells on a plate, while each DNA fragment is copied millions of times by emulsion PCR. So each of these beads with the millions of copies is placed in a well on the plate. There's approximately 1 million wells on the plate. And then the flowing process starts, which means that for nucleotide regions are flowed sequentially over the plate. And every time when the nucleotide flowed is complementary to the DNA in the well, a chemical reaction happens, and this chemical reaction produces a light signal, and the light signal is recorded by a camera. So this is what we get. In titanium we have 200 flow cycles, which means that each of the nucleotide regions is flowed 200 times, which gives us 800 light signal values in parallel for each well on the plate. And these light signals, they are actually what we would call the raw data. And so as they are from the flowing process, they're also called flow data. What you see here is a so-called flowgram for one read. On the x-axis you have the 4 nucleotides in always the same order, T, A, C, G. And on the y-axis you have the flow values, so the light signal strengths. And you can see those very low bars. They come from very weak chemical reactions, which means that the nucleotide in question was not incorporated, while those bars that are around 1, they mean that the nucleotide in question was incorporated once. If you have a bar that goes up to 3, it gives us 3 of the nucleotide in question, and so on. So you can see from that there can be certain inaccuracies, because if you have a flow value of 2.48, that is rounded to 2 nucleotides, while if you have a flow value that is just a little bit higher, 2.52, you will get 3 nucleotides. So this is where over and undercalls occur. And what we did then was that we plotted a histogram. So on the x-axis you have the flow values, on the y-axis you have the frequency of flow values over a whole run on a log10 scale. And you can see nicely that the peaks of these distributions lie on the integers, and that every homopolymer run length belongs to one distribution, or the other way around. And but it is to suspect that these distributions overlap on the sides. So this is where the over and undercalls occur, and substitution errors can only occur if an overcall follows an undercall, or the other way around. So what we did then was we tried to find out how these distributions overlap, and we mapped our data to the reference genome, which was in this case CBOS and also E. coli. And so we got one, every color is one distribution, And we can see that these overlap quite heavily. It actually looks a lot worse than it is because this is again on a log 10 scale, but still this is where the sequencing errors occur. And we can see that these distributions broaden towards longer homopolymer runs. So the green one, the green distribution is a lot broader than the orange one, for example, which simply means that if you have long homopolymer runs, it's more likely to contain an error than short ones. But we can also see that there are some neighboring peaks in these distributions, and we were very unsure where these come from. So this is again a beautifully shaped Mount Matterhorn in Switzerland, and this is more what it actually looks like. Our flow distributions. This is actually Cerro Torre in Patagonia. And so we came up with the idea that these neighboring peaks probably come from real biological discrepancies in the data. For example, CBUS is a diploid, so that might explain it. Or also for E. coli, we knew that our reference genome was not very good. So we came up with the idea that we could use the mate pair runs that we had from COD data and use only the linker sequence, where we could be sure that there would not be any biological difference. And that's what we got. So this is 15 flow cycles for the 42 base pair linker sequence, 1 distribution per position, and we also have the distributions for the negative flow values, which means the flow values that did not lead to a base call. And again, on the x-axis, there's the flow value. On the y-axis, there's a frequency. And the gray areas contain the correct base calls. So everything that is to the left or to the right from the gray area gives a miscall. And we can see that we have some what I would call distribution tails, where we could say that they come from inaccuracies in the pyrosequencing process. But we could also see some neighboring peaks. And we call these putative PCR errors because this is quite a mystery. We don't know how they arise, but they don't look like normal pyrosequencing errors. So they must come from another stage in the sequencing process, which could be the PCR library preparation or also the emulsion PCR, where the DNA strands are copied. So why should one distinguish between error types? Well, the answer is quite simple. So there's a bunch of different applications on 444 pyrosequencing data, and there are some quality trimming and quality filtering algorithms that come with the 444 software, and there's both whole read filtering and read trimming. But the problem is that some of these algorithms are not strict enough, and also they're not sufficient. So people have, especially in the field of metagenomics, come up with ideas how to do additional trimming, and not only stricter trimming, but also designed additional algorithms of how to trim reads from the end and achieve a higher quality and remove errors. But if you want to design algorithms, you have to know what kind of errors you're facing. And especially in metagenomics, the problem is that if you have too many errors in your reads, you will get an immense overestimate of the diversity of species in your sample. So we started with our empirical distributions from CBUS, and the idea is very straightforward. We wanted to count or estimate to what extent PCR or putative PCR errors account for the overall error rate. And what we did was that we constructed intervals around our peaks, which contain the middle 95% of values, so that we could say that if a flow value is known to be from the 1 distribution and it is between 0.5 and 1.5, then it's perfect. It gives us the correct base call. If it's slightly higher, it might be a pyrosequencing error, or also slightly lower. But if it lies in the area of the next neighboring peak, this is very unlikely to be a pyrosequencing error. So we constructed these intervals, and of course 95% gives us a very liberal estimate. So we constructed intervals of different sizes, all the way down to very small intervals, which is a very conservative estimate. So the truth lies somewhere in the middle. And then this might look very confusing at first, but it's actually very simple. So let's take the middle figure, which is for the 1 distribution. If you have on the x-axis the flow value between 0.5 and 1.5, a value will end up in bin 3, which is the green one. It gives a correct base call. If it's slightly higher or lower, it will be counted as a pyrosequencing error, and if it lies in the region of the next neighboring peak, then we will count it as a putative PCR error. So we took our paired end runs from code and just counted how many values fell into each bin, only for the 42 base pair linker sequence. And we actually had a lot of data available, so this is based on 21 mate pair runs. This is millions and millions of reads. So we got quite a good estimate. And well, of course, this is a very big range between 4 and 53%, but as I said, this is based on the very conservative and the very liberal intervals, so the truth will lie somewhere in the middle. So we have discovered a new error type that no one has ever talked about, and there is a publication on PCR errors in 444 pyrosequencing for amplicons, but that's a different kind of error. But they have also discovered quite some high error rates. So we did another analysis which goes a bit more into detail. On the x-axis you have the flow cycle. I was talking about that titanium has 200 flow cycles earlier, which gives us 800 flows and base pair and reads of around 500 base pairs. So the dark blue curve is what we counted as pyrosequencing errors, and the light blue curve is the putative PCR errors. And it is known from literature and from many analyses that reads degrade towards their end. So we did expect the pyrosequencing error curve to rise towards the end of reads. And we can see very clearly that the PCR error curve stays on kind of the same level. So this actually corroborates our theory that these are, in fact, two different kinds of errors. So are there more error sources? Yes, there are. We have adapter sequences that actually should not be reached during sequencing, but sometimes they are. Because when the clones are too short, part or the whole adapter is also sequenced, and 444 does have some algorithm to remove these adapters, but if the adapter sequence is only partly included in the read, or if it contains sequencing errors, then sometimes these algorithms fail. Also, we have artificial duplicates, which make up to 40% in metagenomic reads, and so this gives a very, very wrong estimate on the abundance of a species in a sample. So people who are working on metagenomics are removing these with tools like CD-HIT, for example. Also in de novo whole genome assembly, this totally confuses coverage statistics. So this is a big issue that people are working on currently. Finally, I'd like to thank my supervisors, Kjetil and Inga, who are in the audience, also Frank Nielsen, Anders Lansen, Markus Grome from TH Wildau, Chris Quince from University of Glasgow, Kjetil Jacobsen and the Core Genome Project team from University of Oslo for providing us with the data, and the Research Council of Norway for funding. And I'd also like to thank the organizers for providing me with a travel fellowship. Thanks for listening.